I think we'll go ahead and get started. Welcome, everyone. Thanks again for taking the time. I am James Boyle. I'm from the Fishery Management Plan Coordinator for Shad and River Herring from the Atlantic States Marine Fisheries Commission. Hold on one second. I need to start the recording. Okay, yep. Just a few quick housekeeping things as we get started. Again, if you're new to GoToWebinar, this is our you can raise your hand using the hand raise feature, and please remember to put your hand down. It doesn't automatically go down as it does in some other platforms. Also, everybody will be muted by default, as I just mentioned. If you have not done a mic check, then you are muted both on the organizer's end and on your own end. So if you would like to ask a question later on in the presentation or in the discussion portion, feel free to raise your hand and I'll be able to unmute you on my end, and then you can unmute yourself. And then we ask that everybody please remember to close their mics once they're finished asking a question. We also welcome Jonathan here. Jonathan Watson, as you all know, is my core organizer. Unfortunately, due to the shutdown, he is not working this event, but he is here in his personal capacity, and we're really happy to have him. So with that, I'll show the agenda really fast. One second. Okay, you should all be seeing the agenda. Now I have a very quick update from the commission's point of view. We did not have a Shad and River Herring meeting at the annual meeting that we just had last week. So there's not much to update the forum on. We do have a few sustainable fishery management plans in the works that will be updated every 5 years, and a lot of them were approved in 2020. So as we're closing down 2025, we're getting a lot of those updates in. We should have those for technical committee review and then board review coming up in the next couple of meeting cycles, perhaps. So that's really all that's going on from our end. We also, that's about it. I'm going to pass it off now to Jason Didden for a Mid-Atlantic Council update. Jason. Your mic's open.
Okay, can you hear me now?
Yep, sounds great.
Okay, great, thanks. Okay, thank you. Just a few things to flag from the Mid-Atlantic Fishery Management Council side of things. Again, I'm the Jason Didden Plan Coordinator for mackerel, squid, butterfish. So we had a new Atlantic mackerel stock assessment, and the just base output suggested the stock was increasing substantially. That, you know, taken at just face value, would have suggested potentially some substantial increases for the Atlantic mackerel quota for 26 and 7. And the council a month or two ago kind of approved, at least in the kind of a skeleton of a framework that for Atlantic mackerel would both look at potential expansion of the quota, and then either leaving the river herring and shad cap where it was, or if the quota goes beyond 17,000 metric tons, allowing it to scale up in kind of the ratio method of maybe 5 years ago. So our SSC took a look at that and basically said that the terminal year recruitment estimates are so uncertain that they should be kind of dialed down in the projection period, and more than the range of dialing down that the center performed before the shutdown. So I don't know what the exact numbers are going to be, which makes kind of finalizing the action a little challenging. Council is looking at we have got a river herring and shad advisory panel meeting scheduled for the 21st, and then a joint mackerel, squid, butterfish, and river herring and shad committee meeting for the 24th, going into council action in December. So with the federal shutdown, there's a variety of uncertainties, but principle among them right now is I can't have the center calculate what the new catch limits would be. I anticipate that the quotas that come out will probably be between, I don't know, maybe 5 and 15,000 metric tons for 26 and 7. So those are below the 17,000 metric ton and change that kind of the ratio method would scale up to at the current 129 metric ton river hen and shad cap. So unless kind of my ballparking is way off, I don't expect enough of a mackerel quota to potentially trigger that scaling up of the river herring and shad cap. And we're gonna, the council will be, again, the committee will look at a framework document with a range of alternatives. But at this point, it doesn't look like it looks like there'll be some increase in the Atlantic mackerel quota, but not enough to kind of trigger potentially increasing that river herring and shad cap on it. So that's, again, that's a preliminary kind of just doing some ratio scaling based on the sensitivity analyses that they did have, that they did conduct some past projections from 2023. I think I can kind of eyeball what's going to happen, but not going to know for sure until until Kirsten Curdy at the center is back and can rerun those projections. So the council's plan is to kind of continue moving forward with development of the action, and obviously we get the final. We don't have the final numbers by December, then won't be able to take action. But we're continuing to move in that direction, and hopefully we have that for the December council meeting, and also hopefully they're close enough to my ballparking that the alternatives in the framework make sense. If we get the numbers and they're way different than what I'm expecting, that could kind of have to pause and kind of redevelop those alternatives to match whatever numbers come out of the updated projections, with essentially the SSC turn the dial down on the terminal year recruitment for mackerel. 2024 is a terminal year. The assessment has a history of overestimating that terminal recruitment. And so they are dialing that down from, it was like 1.3 billion age 1 fish in 2024, down just, I think, to about 7% of the assessment's estimated value. That scales the biomass way down, and I don't think it will hugely interfere with the rebuilding trajectory, because last time they also scaled it down. So I don't think it's going to have a huge impact on the rebuilding trajectory. Council did request, right now we're targeting a 61% chance of rebuilding mackerel by 2032. As part of the council's response to the executive order looking at seafood competitiveness, the council said that it would consider a rebuilding plan that just has kind of the Magnuson baseline 50% probability of rebuilding by 2032. So potentially the difference between 60% and 50, between 61 and 50%, those would be two options that the council would have before it to decide. I think it's probably, might be 2,000 metric tons difference in each year between those two options. So anyway, so we're kind of moving forward with an estimate of what that's going to result in, and if we don't get it, or it's wildly different than expected, then we'll have to change our plans. So again, folks, if you go to the council's calendar, you'll see the river herring and shad advisory panel scheduled for the 21st, and then the committee, MSB, mackerel, butterfish, and river herring and shad are going to meet jointly on the 24th to develop recommendations. So there'll be kind of a framework document posted for the certainly by close of business on the 17th for the committee, maybe a little bit earlier. And so folks can track those meetings on the calendar on our website. I think those are the things that I wanted to cover, and I can take any questions if anybody has any.
Thanks, Jason. Yeah, feel free to raise your hand if you have any questions. Also, feel free to use the questions box. I can see that as an organizer. I think in past we've learned that presenters can't see that, but I'm happy to read them out loud if anybody would prefer that. I see, so in the questions box, I'll go to Jonathan first. Jonathan, I don't know if you want to ask your question out loud, and then I'll have Kevin next.
Yeah, thanks, Jason. I was just curious if the bycatch cap can scale up as the mackerel quota increases. Has the council ever discussed the bycatch cap scaling down as the quota decreases? Thanks.
Yeah, it did scale down from it may have been 200 and change when it first started. I'd have to go double check that. So as the mackerel quota scaled down, it did scale down some. I think there may have been a year when it got down to 89 metric tons, but then there was concern that below 129 metric tons, especially at the start of the season when we're transitioning from the prior years. So the way that cap estimate works, it starts off with the prior year's data, and then there's like a kind of an exponential transfer to current year data through 5 trips. So once you get 5 observer trips, you're just using the current year data. And it'd be like 1 trip. There's like a math function that the NIMS uses. You know, at 1 trip, it may go to like 45%, and then 2 trips, 65%. So it jumps to the current year data relatively quickly. But the concern was at a much lower or at a lower cap than 129, starting just getting into more problems with just monitoring that cap and getting enough current year data where you're not just shutting the fishery down based on prior year data. So once, I think there was a year where maybe the quota was around 17,000 metric tons. That was 129. The council is, I think 2 years ago, discussed potentially lowering it or relowering it to 89. Ended up just leaving it at 129 metric tons. Again, primarily due to this concern about, do you let the fishery generate enough data to where you're closing it based on current year data? It's going to be a real problem in 26. I don't know what NIMS is going to do. If they were open right now, I'd probably be talking to them about this a bit already, because there are no directed mackerel trips for 24 and 25. So I don't know what data they're going to use to start the fishery as a assumed rate as they start transitioning. I don't know, maybe they'll take a 3 or 5 year average from a few years ago, but they're not going to be able to use their normal process for that initial rate. Anyway, I started rambling a bit there.
Yeah, that's helpful. Yeah, thanks for popping the hood a little bit and explaining, how that is calculated. I appreciate it.
Yep.
Thanks, and Kevin, I unmuted you on my end, so you should be able to mute now.
All right, am I coming through now?
Yep.
Awesome, thanks. Yeah, and I was going to ask the same question that Jonathan asked there, but I guess Jason, am I understanding it correctly that if the previous year had low catches of river herring and shad, then what transferred over wouldn't be an issue, correct? Like it's only an issue if it's a really bad year where they have a lot of bycatch, then they'd be starting the next year with that high ratio, correct?
Potentially, yeah, if they start off low, then they're not going to get closed unless they start to have some high bycatch ratio observed trips in the current year. And I think, I'd have to double check. I think there's definitely been, I'm pretty sure there was a year where, like it got shut down with not 5 trips. So then the next year was maybe only based on like 2 trips from the year before, because it was like it had shut down mid-transition rate, or at least there was a potential for that to occur. So that's but yes, if whatever they're using as the initial ratio is really low, then it's going to be current year data that shuts them down, the kind of reverse situation.
Gotcha. Yeah, I think my concern is that almost makes it function better if the idea is to incentivize them to avoid areas of high river herring and shad. By actually scaling it down, where we'd be at now would be, they can't afford to get that bycatch. So, obviously I wasn't at the table for any of these things, but it seems to me that for, as us river herring biologists, that to us shows that program would work, because if you're coming in with a high ratio, you don't want to do that. So the idea would be don't catch river herring and shad. If they can't avoid them, that's one thing. I do understand that, but not scaling it down. I think it was 2023. If they had stayed at 89, the fishery would have been shut down. But by moving it up, it allowed them to, harvest more fish that year, which was our worst year, one of our worst years on record. So, hence that's just my curiosity behind that. It seems as though lowering it would, in fact, incentivize them to avoid the areas that it was designed in the first place to do. So, thank you for the background on that. I appreciate it.
Yeah, well, certainly the lower that cap is, the more, the faster, from whatever the ratio is that they would achieve it. That's yeah, I mean, that's just a math operation there. Yep.
Thank you very much. Are there any more questions for Jason? You can put your hand up now or put in the questions box. I'm not seeing any. Feel free, if you come up with them later, to put in the question box. Maybe we can come back to them, or I can contact you with Jason afterwards. Next up, we have the New England Council update from Jamie Cornyn. Jamie, take it away.
Thank you. Good afternoon, everyone. And thank you to the organizers for continuing to hold the forum even during a partial government shutdown. And I have quite a few updates since our last meeting that we had in March, and so hopefully I'll cover everything. The first thing to note was shortly after the forum, there was a peer review of the Atlantic Herring Research Track Working Group's work to develop a new model for Atlantic herring, and that passed peer review. And so there's information about that I can share if folks are interested or want to see that report. Also, since then, in May, I believe the organizers sent out an email blast to let folks know that the New England Fishery Management Council had paused work on Amendment 10 and did not intend to pick that work up this year, and shifted to a different priority. And Amendment 10 is going to be part of our council discussion of priorities for next year, December meeting. So our December meeting takes place in Newport, Rhode Island. There's information about this on our website. And the third day of our meeting, which is Thursday, December 3rd, there'll be a discussion of council priorities at 10:15. The public is welcome to join us in person or online, and there will be an opportunity for comment. One of the main questions will be whether or not to restart the work on Amendment 10, so I want to make sure you're aware of that. So second part is after the forum in March, our plan development team and technical committee from ASMFC worked on a revision to the quota recommendations for the Council for Atlantic Herring. And the final action on that took place in April of last year, and the council submitted it its document to NOAA Fisheries for review in May, and a proposed rule on those quota changes went out, and that comment period closed in July. However, the quota changes were not implemented prior to the shutdown. So at this time, there's two outstanding questions for the herring quotas. Will they increase as the council slightly increase from where they are right now for this year? What will be the quota next year? And will the fishery have occasionally, if there's not a lot of catch in Canada, there are certain provisions where the area in 1A would receive an additional 1,000 metric tons of herring quota, and usually that's in the fall. Unfortunately, that evaluation occurs on October 1st, and we all know what happened that day. So right now, there's a lot of uncertainties about what the herring quota might be for the rest of this year, and then what it could be for the start of next year, given the uncertainty right now in government. NOAA Fisheries is monitoring all of the quotas, and although they're not updating their web pages, they are having actions that prevent overfishing published. Right now, the only area that is open to or doesn't have a 2,000 pound possession limit for Atlantic herring is Area 2, and Area 2 is the southern New England, mid-Atlantic area roughly. And that's the only one right now through the end of the year that has that doesn't have the 2,000 pound possession limit. So lots of uncertainties as we're going toward the end of the year and the beginning of the year. And also just want to let you know the council's work will continue in December to determine which priorities to work on next year. We do have one thing we will be working on for sure. And like I said, in March, there was a peer review of the Atlantic herring stock assessment, the new model, which is a WAM model, a states-based model. That will be updated through the management track process for Atlantic herring in June. And so we'll have, from that model, we'll receive stock status and catch advice. The research track doesn't provide those two parts, but it provides the recipe for determining those. And then the cake, as they say, will come out in the management track. So in June, we'll get that management track result, and we'll be working on an action to set quotas for based on that result. And the council, again, will need to identify what else it wants to work on with respect to herring for next year at their December meeting. So I can take any questions if you have any on what the New England Council is working on this year and next year.
Thank you, Jamie. While we wait for any hands, I saw one go up and then down again, so I'll just wait for a second. I had a quick question. Maybe I missed it, but does the New England Council have a mechanism for written comments as well as verbal comments for the meeting?
We don't have like a form, but folks are welcome to provide written comments on our council meeting page. There is some instructions on how to do so. We take comments up to a certain deadline, and those are compiled and provided to our council. So those written comments are, there's some information on our website about how to provide those, and it's specific for each meeting. So let me look real quick. For the December council meeting, if you go to the actual agenda, it says that comments must be received by Tuesday, November 25th, and it gives you instructions on how to either mail those or email to those and address them to our executive director or our council chair. I'll put the, I'll copy the web link into the question box, and if you want to share that, you're welcome to.
That'd be great. Thank you very much. And I'll make sure when we send a follow-up email after the meeting to send out links for both councils to at least the calendars for those meetings, and then also include these public comment links as well. So thank you for that. I see Maria, you have your hand up. You are unmuted on my end, so you should be able to unmute now.
Okay, can you hear me?
Yep.
Okay, great. Yeah, I'm Maria Abate. I am the research marine biologist for the Wampanoag tribe of Gay-headed Quina. And Jamie, thanks for telling us what's coming up for the council. I had a question though. Did I hear correctly that there will be some discussion about why not to put Amendment 10 back on track? Did I hear that? And I just wondered if you could sort of shed light on reasons that it would be considered better not to go forward, if you have any thoughts on that. Thanks.
Thank you. So the council, when it paused work on Amendment 10, said it would revisit when to restart it during its priorities setting process in December. So when we have the council meeting in December on the third day, so it's a discussion about priorities for the following year. And so this is an opportunity for the council to revisit its discussion and decide if it wants to restart the work on Amendment 10. Without that discussion or motion, the work would continue to be paused. So it would be helpful for the council to hear from the public as well as ahead of time of that meeting to understand what they want. But it's not automatically restarting in January without the council weighing in on the status of that action.
Oh, okay, I understand now. Yep, thanks.
Thank you. And thanks again, Jamie and Jason. If there are no more questions, I think we're ready to move on to our technical presentations. So first up, we have Chris Murray.
Yep, looks good to me.
Good. All right. Feel free to take it away.
So I don't think we have to convince the folks at this presentation that river herring populations have been in decline for several decades, and despite rigorous attempts to bolster the populations, recovery has been uneven, and there's been some really good work done recently to identify the major threats to river herring recovery, and also, what are some key bioindicators that we can measure in order to identify the factors contributing to the lack of recovery. You can advance a few times. These include condition, histopathology, environmental monitoring, and the one I'm going to talk about today is genomic tools. Traditionally, genomic tools had a limited space in conservation biology, but now with the advent of improved technologies and cost effectiveness, they're becoming more widespread. You can advance, please. So what can RNA sequencing tell us? You can advance like 4 or 5 times here. RNA-seq is a technology. It combines both benchtop techniques and also computational biology to quantify the relative amount of messenger RNA in a particular sample. This could be an organism, a tissue, or a cell. The molecule itself, mRNA, is an intermediate molecule between a gene of DNA and also the proteins that actually carry out the function in a cell. And for most genes, mRNA turnover is quite fast. So the sort of snapshot you get during an RNA-seq experiment reflects the current physiological state of the organism. We know that gene expression is affected by genetic differences, and that's why laboratory experiments under controlled conditions are really a very powerful way to employ RNA-seq. But also prior environmental exposures to stressors can leave lasting effects on gene expression patterns, things like temperature shock or exposure to toxicants. And as I mentioned earlier, advancements in both benchtop and computational technologies have really expanded the use of RNA sequencing to non-model organisms, especially in the context of conservation biology. Advance, please. You can advance a few times. So I was brought onto a pilot study that was headed by Ann Peterson at the time at Northeast Fishery Science Center, in which she was able to take samples of opportunity across 4 different rivers in the Northeast. And the hypothesis is that, or the one that we've been thinking about, is that local exposure to certain contaminants might be inhibiting reproductive success. So along with other samples taken at the time, Ann and her colleagues collected river herring from 4 different rivers that were sort of complementary to each other. The Penobscot and the Narraguagus in Maine have a long history of river herring runs and also a long history of pollution. The Penobscot perhaps more contaminated than the Narraguagus. And then the southern reaches of the Northeast United States, we have the Hudson River and the Metedeconk, where the Hudson has a long history of heavy metal contamination as well as PCBs, whereas the Metedeconk is relatively more clean but still has a history of PFAS and other non-point source pollution. Advance, please. So the sample design was pretty straightforward. These are mostly samples of opportunity. both male and female alewife and blueback herring samples from these 4 different rivers on these dates in 2023. So you can see that the sample sizes are relatively uneven, but nevertheless, this permitted us to excise liver samples from these animals and then go through the standard protocols to purify mRNA and then sequence them on the Illumina platform. Advance, please. One of the problems with doing this type of work in non-model organisms is that you often don't have a reference genome built for the non-model organism. You need a reference genome in order to actually count the genes that are expressed. So in this case, to circumvent this problem, we use these short-read mRNA sequences, 150 base pairs long, to reconstruct what's known as a transcriptome. This is a map of all the transcribed genes in river herring livers, species specific to both bluebacks and alewife. And then we use this transcriptome, this map of express sequences, to then get annotation information. So what these genes are and what their functions are. You can advance. This is a quick snapshot of the bioinformatics pipeline for those interested. It's a lot of different tools sort of used in a sequential way. I'm happy to answer any questions about this at the end of the talk. You can advance. This is one of my favorite analyses to do after sort of loading the data and normalizing everything. It's something called the principal coordinate analysis. This is very similar to a PCA in which you look at the gene expression, sort of global gene expression patterns in all your samples, and then you ask the PCOA to ordinate them on a 2-dimensional plane. In this case, the small sort of circles and squares are individual river herring samples. We plot them on this 2-dimensional plane, and the analysis is unsupervised in that it doesn't know anything about the particular origin of the samples. It just places samples with the most similar gene expression patterns close together. In this case, what we're seeing is that the samples from the blueback herring samples from the Penobscot River definitely partition away from the Hudson and the Metedeconk, so north to south. But there's also a main ordination or a main deviation of variance across sex. So this is not surprising, as sex plays the liver plays an important role in sexual maturation. So these are up migrating river herring during the spawning season. So we see a strong signal of this maturation. At least for blueback herring, we're also seeing a significant signal of body condition, explaining some of the variance around gene expression differences. You can advance. We did the same thing for alewife. In this case, we have a more limited number of samples, but the result was somewhat different. And what we see a very clear clustering of gene expression patterns between the Penobscot and the Narragansett. So despite the fact that these samples were taken in, I think, the same week or in a couple of days of each other, and that these 2 river systems are relatively close, these populations have very divergent gene expression patterns. So the males and the females from the Narragansett have a gene expression pattern much more similar than those female fish from the Penobscot River. You can advance. One of the next steps of any RNA-seq experiment is to do something called differential expression analysis. And this is a statistical technique used to identify the specific genes that are being regulated at a significant level between your 2 samples. So in this case, I was interested in knowing, okay, amongst all the blueback herring that we sampled across all rivers, what are the major differences between males and females? What are the genes contributing to this sexual maturation during the reproductive season? And of the 8,654 expressed genes in our liver samples, over 1,800 were deemed as significant. So this plot is known as a volcano plot, where it has log-fold change. This is the amount of gene changes with respect to males and females. And also on the y-axis is log 10 of FDR. This is a p-value that's corrected for multiple comparisons. So larger numbers of the negative log 10 indicate a more significant result. So these circles are the individual genes. The ones that are in red are significantly downregulated in females. The ones that are in blue are significantly upregulated in females. There's a strong bias toward downregulation. So we know that the females are turning off a lot of pathways as they enter, or a lot of normal liver pathways as they enter the maturation season. But in any result, when you have this many genes, you need to know what are these genes actually doing. You can advance. And one of the most powerful ways of doing this is called a functional annotation analysis. And one popular way is by grouping genes according to their gene ontology or shared gene ontologies. So gene ontology is a huge effort undertaken by various groups that assign certain processes to each gene. And this analysis is essentially what's known as an enrichment analysis or overrepresentation analysis, where it finds genes that have shared functions in a cell that are similarly affected by whatever treatment you're interested in. So in this case, which shared genes that have shared functions or pathways in female river herring that are being up or down regulated relative to males. And in doing this, we can identify the specific physiological processes that are divergent between males and females in this case. You can advance. This is the result of the analysis, and these plots are showing on the x-axis is the level of significance, on the y-axis are these particular go terms. So they're kind of relatively enigmatic, but the top represents gene ontology biological processes. So these are a range of processes that are carried out in the cell. In parentheses is the number of genes that are part of that enrichment. And on the top panel is biological processes, the bottom panel are molecular functions. So just two different ways of looking at these shared different pathways. And what the result was pretty straightforward. What we're seeing is a very normal shift in liver function from the normal pathways that metabolize toxicants, but also perform important functions around proteins and lipid metabolism towards maturation. We know that the liver plays an important role in the maturation of ovaries in producing lipids and other molecules that you use to build the eggs. So I'm looking at these plots. These are sort of what I expected to see. So I want to then get an idea of what are the particular genes that are driving these pathways. You can advance. And one of the most important genes in maturation is known as vitellogenin. This is a highly abundant protein that is a part of the yolk precursor that's produced in the liver. It's transported through the bloodstream to the ovary, where it's then cleaved into its individual protein constituents and is used by the developing embryo once the egg is fertilized. And we know that female fish produce high amounts of vitellogenin in the liver during the maturation cycle. So on the plot on the right-hand side, I'm showing the 6 different groups of blue in the Metidicon first, and then the males from the Penobscot, the Hudson, and the Metidicon. The y-axis is a normalized expression in terms of log counts per million. This is a traditional way of showing relative expression. The females are expressing vitellogenin at a very high level, what we expect. When I look at the males, they're expressing it about a half as high as a level as females. So this is the mRNA, not the actual protein. But we know that males, at least in most cases, should not be expressing any vitellogenin mRNA in their livers under any normal circumstances. It is true that some mRNA transcripts or some males might produce some vitellogenin mRNA transcripts, but this seems like quite a lot. And we know that vitellogenin is an important biomarker of exposure to estrogenic compounds or other endocrine disrupting chemicals in male fish. This is a widely studied phenomenon across the world. You can advance one, please. This plot is showing a histogram of the relative expressions of all genes in the blueback herring males in their livers. So log counts per million on the x-axis and the number of genes within each one of these bins. So what we're seeing is that vitellogenin is being expressed in males at the top 98.8 percentile of all genes. So this is highly anomalous. Even if they were expressing very little, we would see this to the other end of the distribution. So something interesting is going on here. You could advance. So then we did the same thing and looked at the vitellogenin expression in alewife. This time it was a little bit different. The only male samples we have are from the Narragansett Oswegas River and not the Penobscot. And the relative expression is quite a bit lower, about half of what we saw on the blueback herring, but it's still there. It's still not zero, and it's still relatively high, closer to, maybe the 70th percentile of all expressed genes. So what we're seeing is that in all male fish across all rivers of both river herring species, we're seeing anomalously high vitiligen expression in livers. And this is an important indicator of estrogenic exposure or exposure to other endocrine disrupting chemicals. You can advance. And this wasn't the only important biomarker gene. Another important protein that's sort of synthesized in the liver, then transported to the ovary in maturing females, are the zona pellucida genes. These make up different parts of the egg membrane. They're sort of upregulated and downregulated sequentially during the maturation process. And river herring have at least 4 different isoforms that I could identify. And other than this isoform 1, which was expressed at very low levels in males, isoform 2, 3, and 4 were all expressed at anomalously high levels in male fish. So it wasn't just vitiliginin. It was also all or 3 of the 4 important zona pellucida isoforms. You can advance one more. Yeah, one more again. So we looked at these genes also in alewife, and we saw a similar pattern as we did for the vitiliginin, in which the isoforms 1, 2, and 3, or 1 and 2, were not expressed at very high levels, even in some of the female fish. But isoforms 3 and 4 were definitely expressed at anomalously high levels, especially isoform 3 in the alewife. You can advance one more time. So what does this all mean? We know that these genes are well-established molecular markers of estrogenic compounds. An induction of VIT, the synthesis of VIT by estrogen, is dose-dependent within broad limits. So if an animal is exposed to higher levels of an estrogen or a chemical that mimics the effect of estrogen, it'll be expressed at higher levels in the organism. We know that VIT is supposed to appear naturally in maturing females and is not supposed to appear in males or immature fish. So the presence in male liver indicates very recent exposure to estrogenic or other endocrine disrupting compounds during the upper migration of these fish. There's a whole slew of potential sources, which makes it very hard to narrow down, from natural to synthetic estrogens. This also includes potentially estrogens excreted by female fish when river herring are really tightly migrating up rivers. I would say this is far-fetched, but something that's been noted in the literature. And there's a whole slew of industrial chemicals, pesticides, especially a range of persistent organic pollutants, including PCBs and dioxins, all of which can induce feminization in fish. So there's some important key unknowns in which we want to take this research further. Does this affect male fitness? And this is the one thing that we really want to know, because just because you're expressing the mRNA for the gene doesn't mean that it's being sort of synthesized into the protein. That's the next step of gene regulation. So we know that mRNA and the use of gene expression tools is a bioindicator, but it's not the end-all be-all. So there's a lot of examples of intersex gonads, reduced sperm quality, even male sterilization and population collapse as a result of overexposure to estrogenic compounds. So right now we have paired histological slides of both testes and ovaries from these fish. We're analyzing them for any signs of intersex or any anomalous features that would indicate increased protein vitiliginin in male testes, which would sort of bring this research full circle. But does this actually compromise reproductive success in wild populations? And then thirdly, what are the sources for these estrogenic endocrine disruption in river herring? It's probable that there's a variety of chemicals that are causing this. You can advance. So the next steps. We really want to integrate this transcriptomic results with the histopathological data. Like I said, this will bring the results full circle and knowing whether or not there's any functional difference between male fish with high VIT expression in their testes. But looking forward and pursuing additional resources to conduct this research, what we really need is a larger sample size, an even sample size of males and females fish from multiple different rivers in one region. And we could also focus on the young of the year. These are the fish that were spawned in certain systems and are out migrating, and whether or not they show similar transcriptomic patterns of their parents during the upriver migration. And as I mentioned, this evidence really needs to be paired with other indices, especially fertilization success. If we know that exposure to estrogenic compounds is affecting fertilization success, we know that this might be an important factor that's limiting recruitment in certain river systems. I'll also say that the development of the reference transcriptome is a real step forward for using molecular tools for river herring. To advance this further, we really need complete genome assemblies for these animals, which is a much preferred reference index over a transcriptome. So with that, I'm happy to answer any questions.
Thank you very much. If you have any questions, please raise your hand. Not yet. Give it a minute. Or feel free to put it in the question box and I can read it out. Yes, Alan, you are unmuted on mine. Now you're unmuted.
Okay, thanks. So is there, maybe this has been done, maybe it hasn't, but is there any work been done looking at whether or not the percent, the sex ratio is changing within some of these rivers? That there's or taking samples of fish to see if they're not genetically showing that they're actually male.
Yeah, I think, sex reversal in fish is something that happens. We scoured the literature for evidence of hermaphrodism in herrings across the globe, and it's very rare. So genetically, sex reversal is not something that you would expect to see in river herring. When you see a testy that's converted to an ovary as a result of exposure to external estrogens or other endocrine disruptors, you don't necessarily see a normal ovary. What you see is some parts of the tissue that are more testy-like, other parts of the tissue that are more ovary-like. You definitely will see maturing oocytes in a sex-reversed organ. So I don't know of any surveys that are looking for examples of sex reversal in river herrings or the sort of intersex gonads that'll be a hallmark of estrogenic exposure.
Okay, thanks.
Yep, Jonathan, go ahead.
Yeah, thanks so much for the presentation and really interesting work. Getting a little bit of feedback here, so I'll try and make this brief. But just a very basic question for you. You said that the mRNA expression you were seeing was indicating very recent exposure to estrogenic compounds. How recent? What's the timeline there? Is it in the last 2 weeks, in the last 24 hours? Like, what? Just curious in terms of where these fish are and when. If you could make inferences about, how long they're in the system or what parts of the system that may be experiencing these exposures, just based on sort of that timeline. Thanks.
I think to really nail it down, you would need to do some sort of controlled laboratory study, because I think that the metabolism of these might vary between species. There are good studies that are done on model fish. I'm thinking of things like zebrafish, mummy chugs, that have actually very closely tracked the timeline. And fish are very sensitive to estrogens, and changes in gene expression will be on the order of hours to days, not like days to weeks. So this is very recent, within hours to days. If we were also able to measure vitellogen and protein in these animals, it would give us a better indicator of how recent it was. Because if they have the mRNA but not the protein, it would be hours. But they have the protein, it might be more on the order of days.
Okay, yeah, that it's really helpful. And I wonder, obviously getting samples from, comparing across rivers is helpful, but if you had a system where you had spotting sort of up and down a large geographic area, it might be interesting. If it's that sensitive, it might be interesting to take samples from different sections of the same river to see, hey, this is near a wastewater treatment plant and we're getting elevated levels here, whereas further upriver, maybe not. I don't know. But yeah, very interesting work. Thanks for sharing.
That's a very good point. I mean, oftentimes you're limited with resources. You have to choose whether or not you want to have a more wide net or hyper focus on one system that has, particular features about it that would make it interesting. Thanks for the comment.
Hey everybody, yeah, thanks so much. You pronounced my name perfectly. Most people, I think that's happened like 3 times in my life, so that's very exciting for me. But yeah, my name is Morgan Pizak, and I'm a Librero postdoctoral fellow at Dalhousie University. I'm starting as an assistant professor at the University of British Columbia here in January, but hopefully I'll be able to keep involved with forage fish movement on the East Coast. Today I'll be presenting about the movement ecology of alewife, but my main work that I focus on is the movement of Atlantic mackerel. So if that's of interest, I can present that in the future, but we've had a massive effort in the past year. I think we've acoustically tagged close to 1,000 mackerel, and so we're tracking their movements on an ocean scale, which has never really been done before. So yeah, happy to come back and present that. But today I'll be focusing on the alewife, and this is a project that's been undertaken mostly by Mark Trudell and his team at St. Andrews Biological Station in New Brunswick there. But yeah, you can move forward, please. Yeah, so this paper was just accepted, and I'm hoping that this QR code is working. But in any case, I just wanted to throw this out there in case you're interested in actually taking a second look at any of the figures that I'm sharing here today. But yeah, it was accepted, I guess, back in August, and it's in TAFs. But there it is for anybody interested. You can move forward. Yeah, so I'm not going to talk too much about the importance and biology of alewife, but I'll just give a quick overview. And so this is a diadromous species. I'm sure we all know that. So they're living in the ocean, and then they move into the rivers to spawn, and then they do that a few times throughout their life cycle. We know that there are forage species right in the middle of the food web, so they're linking the lower parts and the upper parts of the food web. They support really important commercial and recreational fisheries throughout the East Coast of both Canada and the US, and they've been harvested by Indigenous peoples for thousands of years. And so overall, very important species, and they've even been referred to as the fish that feeds all. Next. Unfortunately, the alewife have experienced substantial declines throughout the region. Some of the runs have even decreased by over 90%. So these spawning runs and commercial landings have decreased, as well as a decrease in size. And so some of the reasons associated with this could be overexploitation and bycatch, habitat fragmentation, so extensive dams throughout their range blocking access to important spawning habitat, maybe potentially some dynamics associated with climate change and even predation. And so across these 4 things, I think that there's a really important role of movement ecology. And because the technology hasn't really been there until very recently, we've not been able to tag them acoustically. There's been a lot of work with marking, so not electronic tags. So we're just now coming out with technology that is small enough that we can tag smaller species like alewife. And so this study was a pilot where we tagged some among the first few times. Next. So here we wanted to quantify and characterize mortalities. Since this is among the first times that alewife have been tagged, we wanted to investigate mortalities potentially associated with surgery and tagging, but we also wanted to examine if there were any predation events occurring as well. The next objective focused on identifying migration routes, and then from there, we also examined habitat use and the timing of these post-spawning movements. Finally, we examined diel vertical migration as well. Next. So my study took place in Passamaquoddy Bay, which is within the Bay of Fundy there, between Maine on the south side and New Brunswick on the north side. Next. And so to do the study, we used acoustic telemetry. And for those that might not be familiar with it, I'll give a brief overview. So each one of those dots has one of those receivers pictured there on the right. And these are basically listening stations. So they're kind of like underwater microphones. And so we had really great coverage throughout 4 main habitat types: river, estuary, inner bay, where that PB is labeled, and then the outer bay, which is separated mostly by Deer Island there, and then some coastal sites as well. So I think we had around over 200, and they were strategically placed to capture movements across wide swaths of area. So that would be some of those lined receivers there, where you can see a number of them in a row. And then we had some habitat features, as we called them, targeted as well. So aquaculture sites, seal haul-out sites, and herring weirs. Next. So the way this works is that we surgically implanted alewife with these tags. And so at a dam up by that arrow, we captured them by dip nets. And then we perform surgery to insert the tags. And then after that, the tag, when they come in close proximity with one of those listening stations, the station records who was there and when. And so over time, we stack all of these together across all the individuals and all of the receiver listening stations, and we're able to figure out, are there trends in the movement? Are there spatial and temporal trends and where they are? So that was the main method. And fortunately, the tags that we use here were also equipped with sensors that monitored both temperature and depth. So that's where we're able to look at those predation events and investigate diel vertical migration. But not all acoustic telemetry has those special sensors. So this was a very exciting opportunity with this data set. Next. So here is, I've got a couple of different results here, but I'll go through, I'll start on the right-hand side. So we had 7 definite mortalities with evidence from the acoustic telemetry data, although there was a larger number, I think close to 9 more individuals that didn't complete their migration. And so in terms of mortalities, there was 3 right off the bat soon after release. So those were either associated with transmitters that were not working, or they died very soon after surgery. So as I mentioned, this has not been done that many times, so we're still trying to figure out the best ways to actually conduct the tagging. 3 right off the bat, and that's on the right-hand side at the top of the migration route figure. And then we also noted some predation events as well. And so there were, I think, 5 of those, but I'll double check. It's in the next slide. But anyways, so 7 mortalities. And so we released 32, 26 of them made it to the next habitat type in the estuary. And then after 3 deaths and 3 more incomplete migrations, 20 made it to the mouth of the inner bay. There was another predation event. And then at this point, the tagged fish split. So 8 of them went north of Deer Island and 11 of them went south of Deer Island. And this is interesting just because I think it looks like it's less efficient to go north. It's further to travel. So potentially more foraging habitat on that north side, pulling them that way. Yeah, and then another 17 total make it to the outer bay. So yeah, not all of them finished their migration, but again, not all of, we didn't have definitive evidence saying that there was mortality events for all of those that didn't finish. Next. So here's some really cool plots. So just to orient you, we've got the date and time on the x-axis and then the temperature on the y. And so we can see that for the beginning of the plots, most of them are doing their thing around between 15 and 20 degrees Celsius. And then for all of them, we can see a spike, and that is indicative of a predation event. So the predator actually swallows the tag, and then the tag is recording the internal temperature of the predator, and so that's why it jumps up. And so mesopredators like tuna, their internal body temperatures hover around 25, and endothermic predators like the seal pictured here, they spike around 35. So all of these predation events were consistent with seals. So that's pretty interesting given that we had such a small sample size. So yeah, 4 of them, and it looks like 2 of them were about a month after release, and 1 was really soon after. Yeah, actually, it looks like all 3 of them were about a month after release. So that makes sense as they're moving down the estuary into the inner bay. Next. Just to highlight again the routes, 3 were never detected. We found that the mortalities, most of them were earlier on in the migration, so a lot of them were in the inner bay. We did note that some of these movements were incomplete. As I mentioned, there was no evidence of predation or mortality, but there were no more detections. So that means we didn't see them detected any further south along their migration route. But we didn't find definitive evidence of a mortality or of a predation event. So those were recorded as incomplete. And then again, we saw the split, and that 17 made it to the outer bay. Next. Okay, so here, this is called an abacus plot. And so we've got all of the individual alewife on the y-axis, and then we've got week and habitat on the X. And so most of the alewife departed the river within 1 week of release, so they really didn't spend that much time there. And they moved into the estuary, and they sort of synced their movements. Most of them were present there until week 27. And then after that, they spent the majority of their time in the inner bay. So we can see those long periods of time of green around week 27 to week 30. And they spent much less time in the outer bay. So once they reached that outer bay, they kept moving, and then they departed Passamaquoddy after that. And we had very few detections on the coastal receivers, but that's likely because we had way fewer coastal receivers relative to the other habitat types. Next. So something I want to note is the oscillations. And so this is not the first time this has been recorded. There's a few other studies. So we found that 13 of the alewife demonstrated oscillations between the river and estuary habitats. And so that can be seen with all of the yellow arrows. There were some oscillations between the estuary and inner bay, but much fewer compared to the river and estuary. And then again, more oscillations between the inner and outer bays. And so this has also been found in alewife migrating around Long Island, New York, and then another study further in the Bay of Fundy in the Minas Basin. And so there's a number of reasons that this could be happening. A major reason for oscillations are barriers, but here the system is quite open compared to higher up where there might be more dams. So that's probably not the case. There could be oscillations associated with environmental changes. That could be the case just given that the Bay of Fundy has the most intense tide in the whole world. So they could be waiting until the flows are favorable for movement. I think what might be going on, particularly for the river and estuary, is the oscillations for osmoregulation. So trying to acclimate between the freshwater and the saltwater habitat between the river and estuary. Next. So this is a generalized additive model, and so each of the panels are weeks. So starting with the release at week 25, and then ending where they are departing on week 32. And so the count there is transformed, but the pink means that there's a higher number of unique individual alewife detected at that spot. And I also want to highlight that the dots are the receivers where the fish were detected for that week, just to give an idea of how well this model is capturing these movements. And so at week 25, mirroring the abacus plot, we can see that there's a lot of alewife detected in the river after release. This makes sense. And they're starting to move down into the estuary for week 26, slowly trickling down into the estuary. And then by week 27 and 28 and 29, we can see that consistent use of the inner bay. And so it's interesting that they're moving down towards the islands, separating the inner and outer bay during week 27 and 28, and then sort of circulating and moving using some of those habitats that are further north again. After that, we can see the split of them moving down and exiting the inner bay and moving into the outer bay for weeks 31 and 32. But yeah, the GAM is especially useful for looking at these more fine scale spatial movements relative to the abacus plots, which help with the fine scale temporal movements, but again, they're grouped at habitat type. So this is great for showing it visually within the site. Next. So yeah, our last goal was to examine if the alewife were undertaking diel vertical migration, but as I mentioned, the Passamaquoddy Bay and Bay of Fundy has some of the most intense tides in the whole world. Next. Just to show you, this was me going out sampling. So that was, and I just want you to look at the ramp there. So 8:30, the angle of the ramp was quite steep, and then you can see by coming back after the field day, and I do realize that was a short field day. I'll own up to that. But at the end of the field day, that ramp is basically slack. So that's what we're dealing with on that short amount of time. And before starting this position at Dalhousie, I had done all of my work in the Great Lakes. So I was like, how am I supposed to disentangle the tide with vertical movement? Like, that is so foreign to me coming from freshwater. But anyways, we made it happen with the magic of more GAMs. Next. So here we used, generalized additive model, and we included tide as a covariate factor there. And so the y-axis is the proportion of the water column. So the 0 would be the top of the water and -1 would be the bottom. And then we've got the 24-hour day period. And so we can see, regardless of what the tide height is, that there is evidence of diel vertical migration. We can see that they're shallowest around 3 a.m. So they're up around the surface for 3 to 4 hours in the middle of the night. And then they transit between 6 and noon, moving down to the bottom, where they spend about a little bit more time at the bottom, so 6 to 7 hours at the bottom during the daylight before they move back up at dusk. And so, yeah, these migrations, the transit time was about 6 hours as well. Next. So other studies have looked at diel vertical migration as well. Some of the methods that they used included trawl surveys and hydroacoustics. Interestingly enough, a lot of the studies that have looked at diel vertical migration in alewife have come from the Great Lakes, which is outside of their native range, although it has been observed in marine systems as well. And so it is likely associated with foraging, following of zooplankton, but it could also be associated with predator avoidance as well. Next. Yeah, so what should we do next? As I mentioned, this is a pilot study. We had a small sample size, and unfortunately, it was only 1 year. So it would be great if we could get some more alewife tagged in larger numbers, and then we could compare movements across years to look at interannual variation in movements. As I did mention, we had receivers outfitted at aquaculture sites, but this is a main concern and research interest of Mark Trudell, is looking at interactions and mortalities associated with aquaculture sites. And so to be able to answer this question, again, we would need a larger sample size to actually model trends in predation events. It would be great to also examine the cause of the oscillations. So I don't do anything hydraulics related, but it would be really interesting to see if the alewife are riding the tide and moving out when it is energetically efficient to do so, or to look at predator density to see if they could be causing these oscillations. And then another really interesting thing that has happened in the past year is that you may have noticed outside of Passamaquoddy Bay, we had very few receivers. So some of those receivers in the coastal area, but there's been an extensive grid, I think over 100 or 200, 150. If somebody's on the call that knows, chime in during the question period. But a grid of receivers has been installed throughout the entire Bay of Fundy. And so now we'll be able to examine movements within that larger area and look at habitat use there as well, which is really, really exciting and has not been done on this scale before. Next. Yeah, so just to summarize, we tagged 32 alewife. We found high occurrences of mortalities mostly earlier on in the migration with multiple documentations of predation events. Some of these mortalities were also likely due to surgery. These migrations took 8 weeks, and the majority of the alewife actually that's wrong, south within Passamaquoddy Bay, south of Deer Island, and we noted oscillations between the river and estuary habitats, although the other habitats as well. And we also documented evidence of diel vertical migration. Next. Oh yeah, I've got one plug here. We conducted a literature review highlighting what's known about the marine ecology for American shad, blueback herring, and alewife here. And that was more of a literature review, no empirical data, but check that out if you're interested. And next. And yeah, I'd just like to thank my co-authors and everybody who helped make this study happen. And with that, I'll take any questions.
Excellent. Thanks very much, Morgan. Are there any questions you'd like to raise your hand or put it in the questions box? Yes, Chris, you're unmuted on my end, so you should be able to mute now.
Can you hear me?
Yep.
Thanks. Thanks, Morgan, for the presentation. I just had a question about the oscillations.
Mm-hmm.
I was wondering where the uppermost location for detection of that oscillating behavior and where that occurred on the river, like the uppermost, I guess, for like where they were tagged and where they might come back to. Yeah, thank you.
Yeah, so we didn't look at it in that fine scale, like within each habitat type. That could be done with something like a network analysis. But yeah, where we looked at the oscillations was between those major boundaries and habitat changes, as I highlighted. So yeah, within each habitat, I don't have the answer for that within the river.
Okay, so there's no nothing to show whether or not they went above like Was it before or after the Milltown Dam was decommissioned?
Yeah, no,
The
nothing
study.
to show, nothing to show for that. We did not have receivers above the Milltown Dam.
Okay.
But the data
Okay.
does
Is
exist.
that-
I've just not answered that specific question.
Okay. I count river herring on the main stem of the river,
Mhm.
and so I hear or see presentations about this oscillating behavior potentially occurring, and so I just would be interested if to see if that's occurring and if we should account for that and river herring counts and all that good stuff. So
Yeah, I'm doing a different type of analysis right now for mackerel, and it's called a network analysis, and it basically sees how connected the different listening stations are. And so that type of analysis would be really great to look at to answer that question. I think at this point, with this study being so small scale, we probably wouldn't have like the sample size to look at that fine scale of oscillations, but definitely a really interesting question and something that I'd be happy to look into the future. And I can pass that along to Mark's team.
all right, thank you. I'll just mention one thing real quick too, just for reference. We did a tagging study similar, and we saw more fish that went north over Deer Island than went south into Passamaquoddy Bay. The ratio was probably like 80-20, like 80 going north and then 20 going, because you mentioned like doing repeatability and doing the study again. And so I just wanted to mention that, that type of behavior did occur on when we did the tagging assessment in the bay.
Okay, yeah, that's super interesting. I wonder how much it's changing year to year, or if it's a reflection of our small sample size. But again, many questions that I would be keen to answer with more telemetry studies.
For sure. Thank you.
Thanks.
I did see one more hand, but it's gone back down. Give the person one second if they want to bring it back up. Oh, Alan, yeah, go ahead.
Sorry, I think I actually had it in the wrong position before this whole time. My question is, yeah, and you may have mentioned it, Morgan, but are these fish, assuming they're still at large, how long will the batteries last? How will you be able to detect them in the future? Are there plans to find out if they return in future years, just to get some data on how long they live, et cetera?
Yeah, totally. Yeah. So these tags, I think, were designed to last 6 months. But as I mentioned, the neat part about acoustic telemetry is that it's advancing all the time. And so the major limiting factor is the battery size, but the batteries are getting stronger. So we're able to tag smaller individuals for longer periods of time. Just as an example, we just tagged the Atlantic mackerel, which I mentioned, and the battery life for the tags that we just put out for them that were mostly around 30 centimeters was 2 years. So the technology is there, but for this study, I think it was around 6 months. But we didn't have great acoustic receiver coverage outside of Passamaquoddy Bay, but now there is with that grid that's gone in.
Okay, thanks.
All right, thank you. Thanks again, Morgan. If there are no other questions, if they're not going to put them in the questions box, I see there's one in there, I think, for Chris Murray. Maybe we'll come back to that in the discussion period. But for now, I think we're ready to move on to Shannon Smith's presentation. So I'll just bring that up 1 second.
All right, thank you very much. Good afternoon, everyone. I'm excited to share some work that we did with the Virginia Institute of Marine Science recently, and I'll be talking about blueback herring egress in mid-Atlantic subestuaries. Before I get started, I want to acknowledge my co-authors, Mary Fabrizio and Troy Tuckey. And I also want to thank our funding partners, the Virginia Institute of Marine Science, or VIMS, the Virginia Marine Resources Commission, U.S. Fish and Wildlife Service, and PEO. Next slide, please. All right, well, I'd also be remiss if I didn't give a big shout out and thank you to all the captains and crew, past and present day, on the VIMS Recruitment Surveys program. These folks keep this long-term survey running. And I'll also mention our funding partners here for this program, VIMS, VMRC, and Fish and Wildlife. Next slide, please. So the anadromous life history of river herring is generally understood and well studied, and this group certainly knows that. So adults move upriver to spawn, juveniles recruit and grow in freshwater nursery areas, and then they egress or kind of make a downriver migration to the coastal ocean. Next slide, please. So the kind of general life history we know, a fair bit about, but this juvenile egress period is less understood in large mid-Atlantic subestuaries, which is what I'm going to be talking about today. Next. So we're going to go down to Chesapeake Bay for a little bit right now. And this is a large, highly urbanized estuary. So there's a very high population density in the bay of folks living along rivers and along the bay main stem itself. And this is especially true in the southern parts of the bay, which is where we're going to be today in Virginia. And so this high urbanized kind of population means that there's almost constant permits coming in for surface water withdrawals from the subestuaries, pretty constant permits coming in for various in-water construction projects. That includes dredging, pile driving, kind of shifting of spoils and sediments around. And this is, for dry docks, marinas, boat docks, kind of anything you can think of. And so a lot of this is happening for on private homeowners putting in these construction permits, industry, military installations. And we also have a lot of shoreline modifications and armoring for coastal communities, industry, and for those private homes. And so a lot of this, of course, kind of leads to a lot of disturbance in our rivers and subestuaries here in the Chesapeake Bay. And this can be especially problematic for juvenile fishes, as a lot of these in-water construction activities can cause mortality. Next, please. So it's especially concerning when you have, a fish that is already struggling a little bit, and that's the case for blueback herring in Virginia, as well as in many other states, of course, along the Atlantic here. This timeline is probably going to look pretty familiar. I'm assuming, lots of similar stories among many other states, but we'll just kind of go through kind of what Virginia is looking like. So there were very high commercial landings of blueback herring between the 1930s and the 1970s when we saw that coastwide fishery collapse. Next. You can go one more as well. Thanks. So for Virginia in particular, in 2008, it was made illegal to possess river herring in some rivers, and that moratorium extended to all state waters in 2012. That moratorium does persist today. However, despite the moratorium, we haven't really seen any evidence of rebounding populations, at least not consistently within Virginia waters. Next, please. So again, despite the moratorium and various restoration efforts, river herring populations in Virginia have not recovered. These restoration efforts have included fishway construction, restoration of riverine habitats to try and kind of restore those spawning and juvenile nursery habitats, and the implementation of time of year restrictions. Next. So time of year restrictions in Virginia are overseen, at least in the tidal waters, by the Virginia Marine Resources Commission, and they're implemented in the 1970s. These restrictions focus on putting restrictions on in-water construction activities that may affect spawning runs or other aquatic life. And so because the current focus is on those mainly anadromous spawning fishes, the current window for time of year restrictions in Virginia is the 15th of February to the 30th of June. Next. And although this window does protect our anadromous spawning fishes for the most part, we are wondering if this current window of time also coincides with when those juveniles are leaving from their freshwater nursery habitats. So is this window protecting those juveniles that may be especially sensitive to sounds and sound waves produced from all these construction activities? Are they being protected during their egress period? Next. So to investigate, we leveraged some long-term data from the VIMS Juvenile Fish Trawl Survey. This is a fishery independence survey that employs a multi-species bottom trawl, and we sample monthly at fixed and random stations. During our surveys, we count and measure fish and record water quality parameters at the surface and the bottom. And for this study, we used data from 1996 till 2023. Next. So for this study, we focused on our 3 major subestuaries in Chesapeake Bay in the Virginia portion. From north to south, those are the Rappahannock River, the York River, and the James River. These little white dots on the map here that you can see represent trawl survey stations in 1 month and 1 year. And this is just to give you an idea of the spatial coverage of our survey. Next. I also wanted to give some context, in water conditions between and among our subestuaries here. And this is just to look at average annual salinity. And so as we can see in all 3 subestuaries, and this is from trawl survey data. So the trawl survey does sample that gradient from low salinity upriver to higher salinity down at the mouths of these subestuaries. And I'll note that the Rappahannock River, being the farthest away from the coastal ocean, is also slightly less saline than the other 2. Next. Again, just to kind of look at what conditions are in these subestuaries in general. And this is just showing average annual temperature. So we've got, pretty similar conditions across our subestuaries, and there's not a run-of-river temperature gradient like there is salinity. Next. I'll also note that the flow is highest in the James River, and because it's closest to the coastal ocean, the tidal influence is also highest in this river. All right, next slide, please. So for each of these 3 subestuaries, we examined months that juvenile blueback herring were present, presumably after they left their freshwater nurseries and entered the trawl survey study domain. And we also looked at spatial distribution during egress and their habitat associations during this egress period. Next. We found that juvenile blueback herring were present in all 3 subestuaries across seasons. And we captured them from October to April, so for a period of about 7 months, which was a bit more of a longer time period than we expected to see them, especially when compared to more northern Atlantic subestuaries. We also captured juveniles in very distinct parts of subestuaries during autumn, winter, and spring. And we observed that their spatial center of mass kind of moved downriver with each of these seasons. And we'll go into that in a bit more detail now. Next slide. Thank you. All right, so to look at those that spatial distribution during egress, we estimated monthly mean centers of fish density. And these were averaged across all of our study years. So these mean centers of density were calculated in ArcPro, and they represent the spatial center of mass of the blueback herring population for each month on average. Next. So here's a look at those mean centers of density in autumn. And autumn we defined as September, October, and November. So if we look at the Rappahannock and the James rivers here, we can see that those mean centers were further upriver in the James and Rappahannock, so pretty close to the kind of most upriver extent of our trawl survey domain. And in the York River, we didn't capture any juveniles in September and October. This is most likely because we have, based on our vessel logistics and vessel size, we can only go so far up the York River and up into those 2 little tributaries you see there, the Mattaponi and Pamunkey rivers. And so those juveniles were probably there in those months, but we just couldn't detect them. But one thing I want to point out here is that we captured in September and October, those juveniles were kind of up estuary in the Rappahannock and James. And then in November, we see a bit of a downriver shift of that spatial center of mass. Next. Looking at our winter months now, we saw that mean center, looking at these juvenile fish densities, kind of concentrates in the middle of each of these subestuaries, and we don't really see any directional movement among months, either up or downriver. Next. Finally, in spring, the mean centers of the population were down at the mouths of each subestuary, presumably sort of indicating that the fish were getting ready to exit. Next slide. So here we have those same spatial centers of the mean center of fish density, but now I've overlaid or underlaid a map of average salinity conditions in each season. So low salinity is indicated by the purple and the blue, whereas higher salinity is indicated in the green and yellow. And I want mostly to focus on this autumn season here, and the fact that the mean centers of density in the James and Rappahannock River coincide with low salinities in autumn. Next slide. So based on the months that we encountered blueback herring in these subestuaries, the current time of year restriction window really isn't adequate to protect juvenile blueback herring, because juveniles were encountered in these subestuaries for about 7 months. That's, you know, a significant amount of time that they're potentially exposed to in-water construction activities that may cause mortality. Because the current time of year restriction window starts in February, that's again about 3 to 5 months after juveniles enter the subestuaries. Next slide. We also found that juvenile blueback herring density strongly related to habitat during autumn months. Specifically, we saw greater fish densities when autumn water temperatures were cooler and when autumn salinity was lower. Next slide. So we looked at these using generalized additive models. We had a model for each of our subestuaries. I'm about to show a big panel plot of plots here. So the James River plots are always going to be on top. The Rappahannock River plots are going to be in the middle, and the York River plots will be on the bottom. And you can see the deviance explained by each model on the left there. Next. So here we're going to look at some partial dependence plots. On the y-axis, we're going to have density. So that's the number of fish captured, standardized by area swept by the trawl. And this is on the log scale. And on the x-axis here, we have temperature. And our 3 columns here are the 3 seasons. So autumn on the left, winter in the center, and spring on the right. Next. Focusing for now just on autumn here. So across all of our subestuaries, we saw that we had greater fish densities when autumn temperatures were cooler. Oh, and I guess I probably should explain. So the dashed line is 0. If our blue, black, or green lines are near that, that's pretty much no effect of our covariate on fish density. But here we do see an effect. Let's see, next slide. So in winter, there's not really, a strong sort of relationship between temperature and density here. And spring, we see a similar effect as to autumn, where we found a greater density of juveniles at cooler temperatures. Next slide. So just to sum up, autumn and spring, we saw greater juvenile fish density at those cooler temperatures. Next. All right, now we're looking at salinity on the x-axis. And across all of our subestuaries here in autumn, we see a greater juvenile fish density at lower salinities. You can go ahead too, I think. All right. Winter and spring in the James and Rappahannock, really no effect of salinity on juvenile fish density. However, in the York River, we saw that we had a higher density of juveniles at higher salinities in winter and spring. Next slide. So the main takeaways here. In autumn, we saw greater juvenile fish densities at lower salinities. Winter and spring, we didn't see a consistent trend across the subestuaries. Next. So based on these generalized additive models and our kind of association with finding blueback in those lower salinity waters in the fall, autumn in Virginia may be a really important osmoregulatory acclimation period for these juveniles that have presumably just left their freshwater nursery areas. Low salinity waters may limit osmotic stress and energetic expenditure for these fishes, and they may be kind of gradually acclimating to these higher salinity waters in the fall. Next. In addition, kind of defining these habitat associations across different seasons for juveniles prompts some considerations for our time of year restrictions in Virginia. Flow conditions that affect both salinity and temperature in our subestuaries will most likely affect where juveniles are in the subestuaries, and that can affect kind of the spatial and temporal considerations for our time of year restrictions. Next. So just to kind of close out with some considerations and challenges. So an expanded time of year restriction window may protect juvenile blueback herring and other elocids and perhaps other anadromous species as well. So again, currently that restriction window is February to June. If we expand that to include a couple months earlier in the year, we may be, protecting those juvenile blueback herring during their downriver movement, during that egress period, and protecting them from some of those potentially harmful construction activities. We also show relatively strong support for applying these restrictions on a case-by-case basis outside the time of year restriction window. And so currently these restrictions are on a case-by-case basis. And again, this just kind of shows the importance of that. And in this way, we can protect both spawners and juveniles year-round. Next. So logistical challenges, the high spatial and temporal variability in habitat conditions and how fish associate with habitats at different times makes the identification of dates for a time of year restriction window very difficult. And again, this kind of comes back to the strength of looking at these on a case-by-case basis. Next. All right, that's all I've got. Thank you very much for listening. And this work has been published relatively recently, and I invite you to check out our paper, and I'm happy to take questions here or later offline. Thank you.
Thank you very much, Shannon. Do we have any questions? Feel free to raise your hand. Yes, Chris.
Hi, Shannon. Thank you, Shannon, for that presentation. Just had a question about the low salinity limiting the osmotic stress. I was wondering if juvenile fish are stuck above dams, do you think that's better than, not better, but do you think that's okay for fish for a certain period of time? And how much time do you think? Because we see it happen all the time until we get huge rain events. And I'm just wondering if it causes stress to be in the upper watershed, but it's sounding like maybe they stick in like more within freshwater environments to limit stress.
Yeah, thank you. I probably should have specified the trawl survey domain. There aren't any dams until quite a bit further upriver, so we're only sampling kind of downriver of a lot of those large dams. I mean, like you said, it's probably not it's not better. I think there's in the 3 subestuaries that we looked at here, at least on the main stem, there's quite a few river kilometers of fresh water that's available to them that is still downstream of dams. And the osmotic kind of hypothesis is speculation on our part for this system and this species at the moment.
Mm.
See, I'm not sure I actually answered your question. You can.
I guess because I was thinking that like them staying in the freshwater environment for too long was going to cause stress, but maybe you were saying that the lower salinities might limit the stress anyways, and that the delay in them being in the fresh water might not be as bad as I'm thinking. I don't know. But they do get out eventually. We just need rain, you know, and sometimes they have to wait a long time.
Right, right. Yeah, that's definitely something that we'd like to look at a little bit further, is kind of get more into the physiological aspect of it. I think based on what I've read and what you've said, I think, you know, if they do remain, you know, in fresh water, that's not necessarily a good thing, but I think it's that gradual acclimation that, you know, when you get down to that tidal fresh mixing area, I think that might be advantageous. So they're staying in those kind of lower salinity waters for a little bit longer when they can.
Mm-hmm. Thank you.
Thank you.
Next I have what I think is Wilson. I think you're up next.
Yeah, thank you, James. So Shannon, great presentation. Thank you so much. I wanted to ask you if I was interpreting your graphs correctly, which seemed to show that you're finding the fish there through the winter and spring months. And is that an implication that they don't really outmigrate from the estuaries until the following spring after they were hatched?
Yeah, good question. So based on the analyses that we did, again, those were kind of monthly mean centers of fish density averaged across all of our years. We averaged across years because the trawl survey, you know, has a limited temporal sampling scope. We do sample every month, but probably only 2 to 3 days in each subestuary. So we kind of averaged across years to get a more robust estimate of kind of, you know, where they were in the subestuaries across those seasons. One of the kind of considerations of this, you know, mean center analysis that we used is that, you know, I can't really definitively say whether those mean centers made up, you know, whether that's sort of a single outmigrating cohort, if you will, kind of a single group going down estuary really slowly, and if they do stay until April and May, or if perhaps our data, you know, our trawl survey was encountering them, you know, separate groups that were moving rapidly down estuary, if that makes sense. And so we need to better or kind of finer temporal sampling would help us get at that question a little better, as well as, you know, different kind of tracking methods and telemetry methods. So, you know, right now it looks like whether they're moving quickly down estuary or whether they're moving slowly down estuary, we are encountering juveniles in all these months, but how long they're actually staying there, we can't say.
Yeah, thank you. That makes a lot of sense, that there could be different cohorts that you're looking at and giving you the center of distribution datum. Are these specimens by any chance archived, and would it be possible to look at the otoliths from any of them? Because that would tell you then maybe if you were, you know, looking at different cohorts, if there were different age groups that you were looking at, like, in other words, some that were spawned very early during the spawning season and then others that were spawned later during the season. So that might get to answering that question of whether the center of distribution is the same or a different group of fish.
Right. Yeah, unfortunately, the specimens that we have are not archived. I did neglect to mention that we only looked at age 0 or young of year fishes, and so we applied a kind of a length threshold or a length cutoff. And we only looked at fish, you know, fish captured from September to May. And so that length cutoff helped us probably make sure we were getting mostly young of year fish. The length cutoff was based on length age study done by the trawl survey folks.
Okay, thanks. Yeah, that should help to to sort some of that variability out by using that length threshold. Still very interesting, and it would be really interesting to know if the same fish, you know, are overwintering that long. I mean, my perception from the past literature, at least for here in North Carolina, is that the fish were there once they respond in the spring. They hung out in the nursery areas and then left in the fall. But your data suggests it might be otherwise. So interesting.
Thank you, Wilson. We also have Thomas Nelson. Thomas, you're unmuted on my end, so you should be able to unmute now.
Hey, thanks so much, Shannon. Really, really cool talk. This kind of gets back to the previous question. So we've got yearling fish, so 1-year-old or sub 1-year-olds that have been collected in the Potomac during spawning, like with spawning individuals. Kind of getting back to some of Karin Lindbergh's anomalous migration things. And so I think there's a chance, depending on the system, depending where they are, that these fish could be overwintering in our rivers and our sub-estuaries. So I really hope, Shannon, I don't know if there's an opportunity to catalog some of these from the trawl survey, or to get our hands on some otoliths, because we are getting ready to run those otoliths to see if those fish ever left or if they've been hanging out overwintering. So I'd love to grab some stuff from VIMS as well.
Yeah, that would be just a great kind of knowledge gap to fill for these rivers. So yeah, feel free to reach out. I'm not at VIMS anymore, but I still talk to my co-authors and would be happy to put you in touch and see what we can do. That'd be great.
Awesome. Thank you so much. Thanks for an excellent talk too. I was very closely listening to this. So it's cool. It's cool to see this data. It's cool to see the juvenile fish are there for what I would say longer periods than the literature would suggest.
Thank you.
Yeah, Jonathan?
Oh yeah, thanks. And great presentation. Yeah, really. I appreciate how you guys, tied it to management through this specific discussion on the time of year restriction piece. My other, taking a look at that temporal aspect, my other question would be, was there any interest, or is there any future interest from you or your colleagues to look at predator relative abundance, blue catfish obviously being a very hot topic in the Chesapeake right now, and looking at, sort of co-occurrence at different times of year with those predators that are around, and making any inferences about, predation susceptibility for those young a year would be interesting. Curious if you guys have talked about that or have any insights.
Yeah, thank you. That's a great question. We haven't directly talked about that, but yeah, that would be a great avenue for future research here for sure. Yeah, because blue catfish are, like you said, a very, very hot topic, and they tend to eat a lot of things. So
Thanks very much, Shannon. We really appreciate it. I don't see any other hands, so I think we'll move on to the next one. Thanks again. And then the next one is Sophie Chivers.
So hey everyone, thanks for the slot to chat with y'all. I'm Sophie Chivers. I work on the Gulf of Maine River Herring Network with Manomet and Maine Center for Coastal Fisheries, and I started working with them on this project a couple years ago while I was a student at College of the Atlantic. And this presentation is a summary of our work on river herring stewardship in Maine. Next slide, please. So as I'm sure many folks here are aware, river herring sometimes struggle to make their way to spawning habitat due to natural and anthropogenic barriers to passage, be they water levels, dams, culverts, or beavers. On many runs in Maine and some in other New England states, community members, volunteers, harvesters, and nonprofits have taken on the work of stewarding these populations, restoring passage, monitoring the populations and environmental conditions, educating other community members, and more. Based on a series of conversations with these stewards and state, federal, and interstate managers, we set out to understand what stewardship entails, who's doing it, how much time they're spending, and what barriers they're running into along the way in hopes of better categorizing the role that stewardship plays in maintaining river herring populations in Maine. Next, please. So our methods are three-pronged. We started off by doing 13 key informant interviews with researchers, nonprofit staff, and state agency staff in Maine, Massachusetts, and Connecticut. These interviews were geared toward providing a coast-wide and multidisciplinary context for river herring stewardship in the Gulf of Maine, and they were sort of designed to inform the protocols and interview questions for the next step, the steward interviews. Next slide, please. So these steward interviews were conducted with state agency staff, volunteers, nonprofit staff, and harvesters who steward a particular run. And the questions really focused on the day-to-day reality of stewarding river herring in a particular place, the things that make it possible, and the challenges that stewards are facing in doing their work. Next slide. The final part of our methodology was a written survey. Anyone who was invited for an interview received this survey link. 16 respondents completed that survey. And this was designed to really quantify the time commitment of stewardship and capture the breadth of activities that stewards engage in. Next slide. So here's a quick sense for the geographic spread of river herring runs in Maine. All of the triangles represent commercially harvested runs, and the circles all represent runs without a commercial harvest. Any shape that's shaded blue represents a site that we interviewed a steward from. And we tried to interview stewards from across the state at a rate that's somewhat representative of the relative number of runs in that region. Next slide, please. So what did we learn? Next slide. One of the key ideas was that stewards do a really wide breadth of activities, both kind of really visibly in the field and less visibly. These activities include passage restoration, not only major projects like the bottom photo, which shows a pretty major dam removal, but also yearly or even daily adjustments like the one happening in the top picture, using sandbags or gates or removing beaver dams or other blockages to maintain and improve passage throughout the season. Next slide. Monitoring is also a really significant category of stewardship activity. These two photos show two different kinds of monitoring. The top one shows fish counting, and the bottom one is of someone taking scale samples in their kitchen. These are both really important and common forms of monitoring, especially on runs hoping to establish a commercial harvest. But some stewards may also be taking environmental data, like temperature or stream flow or rainfall, juvenile data, or monitoring zooplankton populations. Next slide, please. Stewards also reported engaging in lots of activities away from their runs or that are a little bit less obvious, but just as important. This includes education and outreach activities like working with students of all ages and adult community members, organizing community events, attending meetings about monitoring and networking, or participating in town meetings, as well as engaging in broader state and federal level policymaking and coordinating volunteers on their run. So you can see here a couple snaps of some education activities happening with little kiddos, community activities, kind of counting, and then a monitoring workshop that the network recently held. These are all great examples of this kind of less obvious stewardship. Next slide, please. One of the other important learnings we had is just how time-consuming stewardship actually is. So we asked stewards to estimate, or the lead steward on a run to estimate the amount of time that they're spending. And they reported spending a total of between 35 and 296 hours annually taking care of their runs, with a lot of this time concentrated in the spring. So here we've got a box plot of the amount of time that stewards estimate spending on each activity in a year. On the x-axis is the total number of hours spent, and on the y-axis are each of the activities. The red bars are field activities, and the blue bars are those non-field activities. On the whole, field activities were the most time-consuming. Counting adults definitely leads that, with biological and ecosystem monitoring following. And stewards also reported spending a lot of time planning and coordinating stewardship activities. Next slide, please. We also learned that there are lots of different components that make stewardship work. One of those components is collaboration, and respondents pretty consistently reported the importance of partnership. So here we've got a bar chart that shows the primary partners stewards work with on restoration projects and in general. The y-axis represents the number of times a category was reported, and the x-axis represents the different kinds of organizations or partners. Those blue bars are partners in specifically restoration projects, and the orange bars are partners in overall stewardship activities. So you can see that NGOs or nonprofits play really significant roles, as do state agency staff. All stewards reported working with at least one other partner, and nonprofits play a pretty major role in supporting stewardship around the state, providing grant writing capacity, staff time, expertise, materials, and even stipends to stewards. Agency staff also play pretty significant roles adding capacity and monitoring runs across the state. Next slide, please. Many respondents also discussed the importance of commercial harvest in stewardship. This quote is from one of our interviews, and it illustrates one potential role that harvest can have. So the steward said, you need to have a commercial fishery to do the stewardship because you need an incentive because people are not going to do this forever for free. This quote gets at the importance of harvest for motivating and enabling stewardship. Harvest can fund stewardship both by providing income for the steward, compensating them for the time they spend doing that kind of stewardship work. And also because often a lot of that income gets funneled back into the run for passage restoration or counting sites, viewing platforms, things like that, sandbags even, as sort of an investment. And this is pretty important because, as we'll talk about a little bit more in a second, motivation and funding are two pretty major barriers to this kind of stewardship. Next slide, please. So as we started asking questions about challenges and barriers, we realized that in some cases, what serves as a barrier in one community might actually be the key to success in another. So one really notable example of this is community support. Next slide. In some communities, the general public really supports the stewardship work. This quote is from a steward who feels really supported by their community, saying, boy, they've been nothing but good. I mean, honestly, we've never had any resistance whatsoever. I mean, it's been amazing. This really isn't the case in every community. In many places, stewards feel like there just is significant resistance to alewife reintroduction, or in others, the community just isn't very involved. Next slide, please. This quote is an example from one steward saying that they had sent out all these emails begging people or asking for help with counting, and that they don't understand why they don't have more support. So this happens for a couple different barriers, but community support was an obvious one. Across the board, stewards are worried about a couple of different things. Next slide, please. And one of the major ones is continuity and capacity. Many stewards are volunteers or harvesters, and a lot of them are getting older, and they're really worried about who's going to step in after they step back. This quote from the interview illustrates that challenge, saying there were a few folks that were interested in being involved, but no one wanted to be the real champion for the alewife run. And whenever you hear about local initiatives to restore a river herring run at a local pond, you know there's some local champion there. So this really gets at this idea that having a local person or people who really care about the run is really important to stewardship success and longevity. Stewards also reported concern about finding funding and resources. Grants often overlook long-term post-restoration monitoring, management, and stewardship, especially those that are sort of designed for major restoration projects. And it's difficult to find grants that are geared towards this sort of never-ending stewardship process. So finding funding to cover that day-to-day work, pay for the materials needed, compensate people for the time they spend is a challenge. Next slide, please. So as a quick recap, stewardship is a broad set of activities that takes a lot of time, takes a team, and requires a champion. And stewards reported challenges including lack of support from their communities, continuity and capacity, and lack of funding. Next slide. And then just as we wrap up here, I want to make a couple of acknowledgements, mostly to my wonderful collaborators, the Sea Grant Biennial, which funded this work, all the wonderful folks that let me use their photos, and I'd be remiss not to say a huge thank you to everyone who talked to us for this project. Next slide. And then just before we jump to questions, I wanted to share a couple of resources from the Gulf of Maine River Herring Network. That first link is our website, which has lots of information about river herring in Maine and in the Gulf of Maine. And then the second link is to a count portal that visualizes various different counts across the coast over the time period that stewards have been collecting that data. So 2 places to go for more information. And I'd be really happy to take any questions at this point. Thank you.
Thank you very much, Sophie. Are there any questions? Yeah, Wilson, go ahead.
Yeah, thanks, James. Sophie, great presentation on an aspect of river herring management that I think has been little studied. So thank you for doing that. And I will just express my envy at the fact that you all have much clearer water in New England and Maine in particular than we do down here in the Southeast. It's hard to generate enthusiasm when you have turbid or very dark tannin stained waters and you can't see the fish. So you guys have a huge advantage up there, and I am envious. It would be really interesting to try and start a similar stewardship network here in the South Atlantic, and I would love to see somebody do that. Thank you.
Yeah, thank you. Yeah, our water quality or our water clarity is definitely an advantage. That's something we hear from stewards on runs that have more turbidity or more sort of white water, that getting people excited about fish they can't see is really hard. So I totally sympathize and would love to see that work kind of happening further south.
Are there any further? Oh, yeah, Jonathan.
Sure, I'll ask one. Great presentation, and thanks for coming to the forum. You mentioned in that sort of quote, you highlighted that, you know, people aren't going to continue to do this work for free, and you sort of highlighted the importance of commercial harvesters in maintaining the stewardship. Are you aware of models whereby, you know, those runs, and this may be a ignorant question, but those runs that are commercially harvested, there's a, you know, a fee or a tax, and those where those dollars go directly back into supporting stewardship activities? Is that a model that's practiced, or is it more just the community awareness through commercial harvesting that sort of reinforces the stewardship piece?
Yeah, that's a great question. There's a couple different, so in Maine, river herring are sort of co-managed at 3 scales. The ASMFC, obviously the state, and then municipalities have some amount of say over how fisheries resources get used and allocated. Typically, a municipality, once it's been granted harvest rights by the state, will put that harvest out to bid. And so that fee money comes back to the town, and in some cases they use that to support harvesting equipment, in some cases they use that to support river restoration, and in some cases it sort of finds its way back into town coffers and covers other kinds of activities. In almost every case, at least that I'm aware of or have sort of chatted with a harvester, they're spending money out of their own pocket to make sure that fish are making their way upstream. So that might be buying sandbags or gates. Often big restoration projects like dam removals or things like that, I'm sure you're all super aware of how expensive those can be. So those aren't typically things that are coming solely from river herring harvest. Harvest can contribute. Often those are grant funded activities. Yeah, in some cases, there's sort of a community group that works alongside the commercial harvester and does a little bit more of the stewardship and community engagement. And the harvester just sort of takes care of what's required to maintain that commercial harvest. In other cases, the harvester is sort of the primary community volunteer that's really engaging school groups and things like that and creating a little bit more of that community awareness. But there are definitely instances where revenue from harvest is either through the harvester or through the town being looped back to maintain passage. Does that answer your question?
Yeah, absolutely. Yeah, and yeah, maybe, yeah, I guess it's a question municipality by municipality, but I'm wondering, it may be a good chance to highlight the particular municipalities, like, you know, sure that the funds for these commercial runs are, you know, you could, as a community member, perhaps you could advocate that they be more fully reinvested into stewardship activities rather than, you know, going to the general fund or something like that. But yeah, interesting concept, and certainly like the idea of sort of financially reinforcing stewardship through commercial harvest. I think it's a great, you know, multi-pronged benefit there. The only other thing And this is just a comment to highlight. I'd have to look at the agenda again. Maybe Emily from Manomet is going to present on the portal, but if not, I know in past meetings of the forum, we've had presentations about the portal, and just wanted to remind folks, especially state folks or others who are collecting data, that there is opportunities to share your data through that portal. And if you're interested, I'm sure that James can send out he will send out the YouTube page, which it's got past meeting recordings, and we can highlight which meetings Emily presented on that portal and how you can go about getting your data incorporated there. So just a pitch for the portal. Thanks.
Yeah, absolutely. I think there's even a little button. If you go to that app, there might be a little button to say, share my data here. So an easy way to upload it right through the app.
Thank you. Thanks again, Sophie. Thanks, Jonathan. That's a great note. I'll make sure to include that in the follow-up email. I don't see any other questions. And also, for the sake of time, I think we're ready to move on to the last technical presentation. We have Dan Stitch.
All right, I'm going to jump right into this. And James, appreciate you reaching out to get an update on this work. Folks haven't heard enough about it already. I'm going to go ahead and talk about modeling shad and river herring populations yet again. This is kind of exciting, though. A lot of this work is wrapping up for these species, following up on the 2024 stock assessment, and a bunch of really good work by Sean Snyder and others. And so go ahead right onto that first slide. I won't belabor this too much. I'm guessing most of this crew is pretty familiar with the life history of these organisms, but I just want to throw out there that I'll primarily focus today on alewife and blueback herring. Of course, you know me, there's always something to do with American shad hanging out in the back burner. If we could go on to the next slide. I am going to pause for a moment here just to kind of cover the life cycle, partly because this is what it makes it a little bit difficult to create large-scale population models for species like river herring, as well as a number of others when we start to throw dams in the mix. The view I'm going to take on this is a relatively reduced view, the figure here from Hera et al. 2021. I'm going to take this view largely because it's how we're modeling these populations still within the tools that I'll talk about today. So we're dealing with the scenario where these fish are hatched in rivers. They all migrate out to the ocean in one season. None of this back and forth in the estuary. I'm joking around. We're just not modeling it yet. They head out to the ocean, live there for a number of years before returning to spawn, and then to varying degrees depending on the species, and in the case of American shad, depending on the latitude, they'll go back and forth between the river and the ocean for a number of years. So this broken up life cycle makes it a little bit difficult, especially for data poor and data limited species like these, to put together sophisticated stock assessment tools. And I think a number of folks here are familiar with that. We need to break up their life cycle a little bit in order to model it effectively. Go ahead to the next slide. River herring is important. We're all on board with that. For the sake of time, I think we're just going to move on to the next slide. And one more. Cool. And so the big remaining threats to restoration for these suite of species really have come down to things like the impacts of dams and freshwater habitat systems, climate change, and then threat number 3 is mullets, as pictured in the bottom left corner. I'm joking, of course, it's fishing pressure has the potential to also exert influence on these species. And so those are the kinds of things that we're setting out to model as we put together population models for these species. Go ahead to the next slide. So we set out with some basic objectives for every given one of these species, shad some time ago, river herring more recently. And the basic idea here with the most recent take has been to go out and try to quantify river herring habitat. This is building off of the NOAA Habitat Conservation Plan. Then relate that habitat data, those habitat data, to population metrics of interest for both alewife, blueback herring, assess the potential for restoration that's out there, and then thinking about fish passage or fishery mortality rates, start to ask questions like how much is enough? Next slide, please. To do this, we've been building out some now relatively large R packages based mostly on data from the published literature or from the recent stock assessments for American shad and river herring. This is an R package that's out there, open source, free to install. You can find it with a quick Google, but the link is also provided at the end of the talk in the PDF. The R package itself recently underwent peer review this summer, and we got some really good feedback on things that could be improved, and most of that's been integrated. There are a couple of little touch-up things that I'd like to keep working on here, and I've still got emails pinned in my inbox from the GitHub issue tracker for that journal review, but it's more or less set up and rocking and rolling and ready to go. Go ahead to the next slide. And we're going to take a basic look at how that works. And so when we set out, we're looking at trying to model a variety of populations along the coast that may or may not follow, the sort of predisposed notion of what we think their populations should look like. And so we try to make these relatively flexible as we go. This is just an example of an imaginary river in our case, where we've got various outlets connecting to the coastline. Those are in green, and then we've got reaches of freshwater habitat upstream of those with varying numbers of dams going from the bright red to the dark red. Here's just thinking about first, second, third order dams in the system. Sean Snyder has gone through and based on some pretty straightforward rules, maybe not, biologically the most impressive set of rules, but it's a starting point, has gone through and characterized this for the entire east coast of the United States, now Canada as well, and what we think is historical river herring habitat. And some of you might have been involved in that process. We showed up with markers and printouts of all of these maps to the stock assessment and had folks tell us which things we had marked as habitat that weren't, or areas of habitat that we had missed. And we've gone back and forth in discussions about this, but he was basically working on some rules having to do with width of the river, slope, elevation, and length. And it's got all of these rivers on the entire East Coast broken up like this. There are also chunks of habitat that might only be accessible or suitable for particular species. So for example, in River B in the middle of our screen here, we've got a pond that is located right in the middle of that upstream reach. That pond would be great habitat for alewife, but maybe not so much for blueback herring and American shad. So we're trying to account for those kinds of things as we go along as well. And then you'll also notice in that same river, there's a bit of habitat that's inaccessible. One local example for myself and others are upstream reaches of Hudson River tributaries that have waterfalls on them, for example. And so those get knocked out of that as well. And so we start with a really big picture habitat. We start to winnow down and see what we've got left. Next slide, please. And then we're going to go ahead and hook that up to a pretty typical data limited type of population model. A lot of that revolves around estimates of von Bertalanffy growth parameters from otolith or scale data. And so for the benchmark stock assessment, we were able to lean on both of those data sources by sharing information across them and characterizing the differences between scale and otolith assigned ages, so that we were able to maintain a large number of rivers overall, or stocks, whichever you like, in order to inform those parameter estimates. Here, I think we're showing parameter estimates for, oh my goodness, I have to do the multiplication. I'm guessing alewife based on the number, but it could be blueback herring. This set of estimates is just for the males. There's another set just like these, otoliths and scales, just for the females. And then we have sex aggregated growth curves. All told, it ends up being somewhere around 500 or 800 sets of growth curves for the fish that are feeding into these models. The cool thing about this is that we're able to break this up by region into different life history or genetic reporting groups for alewife and blueback herring, and then use those estimates to make predictions on finer scales around the coast. We use these growth parameter estimates to inform mortality estimations. So Wes Aiken did age-specific mortality estimation for those, and then he and Katie had pulled up a stochastic per recruit model, and we took routines from that and maturity routines from the stock assessment, and we balled this all into the R package in order to make this population model work. And then to get it all to hold hands, we apply carrying capacity in freshwater of some number of adult fish per spawning fish, per acre of surface area. And for blueback herring and alewife, that's considerably higher, as we have talked about for American shad. And there's actually a little bit of data out there from modern times to support those numbers. Let's go ahead to the next slide. And so I've shown this figure before. This one's from the American Shad paper that Joe Zedleski read. Sean Snyder has a paper in review right now for both alewife and blueback herring and frontiers. And it's kind of taken a really similar approach. In fact, these are the scenarios that we used for the 2024 ASMSC River Herring Benchmark Stock Assessment. So we're going to consider 3 scenarios here just to try and take a really gross 60,000 foot perspective look at things. We've got a completely undammed scenario where we can go down into each of these rivers and pull dams from the rivers. We've got a dammed scenario where we're going to imagine for a minute that the fish can't pass any of the dams and any of the rivers. And then we're going to go ahead and make an attempt at characterizing what we think are current passage performance for these species at dams. And that's there's an asterisk on there. It's not real well characterized still. There's some good research that's been done in recent years. But we still don't have a great handle on what either upstream or downstream dam passage looks like for most of these species, getting better all the time. For this case example, I think we use very similar passage standards to the shad paper, somewhere around 30% per day upstream average and a 85% downstream for adult, 95% downstream for juvenile survival through dams. So we'll go ahead and we'll compare some results and some projections along these lines in the following slides. Nice job picking up on that cue. First thing we did was we went ahead and characterized the amount of habitat that's out there. This map may look really familiar to some of you who worked on the Habitat Conservation Plan. It's because it's mostly the same hucks that we're working in. These are scaled just a little bit, I guess, larger than those Huck 10 watersheds that we were thinking about in that case. And the reality is we can kind of work anywhere in between this and an arbitrarily defined watershed at this point. A couple of big picture trends to consider here is that alewife habitat penetrates just a little bit further inland than blueback herring habitat in most of their shared range. The other big trend is that blueback herring habitat persists a bit further south than it does for alewife because they live further south than alewife do. The last thing that might pop out at you, depending on how familiar you are with these systems, is the noticeable absence of the upper Delaware and Susquehanna rivers in there for blueback herring. After discussions with folks, both during the River Herring Forum, during the stock assessment peer review process, we've actually gone ahead and added those systems in for bluebacks so that folks can play around with those data to their heart's content. Let's go ahead to the next slide. This is another way of looking at those same data, and this time, instead of just looking at the Huck 8 watersheds, we're looking at the order of dams within each of those. The segments that are yellow have very few dams, that are 0 to 2 dams there. Orange segments, slightly more, 3 to 6 dams. And as the color of the red intensifies, you're approaching upward of 24 dams. And you can see that a lot of those are distinctly positioned in southern to central New England, right? And so you've got small swaths of habitat that are largely inaccessible in those regions, whereas in the mid-Atlantic region, a lot of that habitat is a lot more accessible, and that's going to be important to kind of guide how we think about the results of the population models. On the next slide. I lied. This is just breaking the habitat up into the genetic reporting regions from Carey's paper a couple of years ago. So I'll report the results in these same groups. There are 5 of them for blueback herring. Southern, we've got South Atlantic, the Mid-Atlantic, Southern New England, Middle New England, Northern New England, Canada, and then those last 3 are the same for alewife. Again, they just don't go as far south, and they don't break out distinctively in Southern New England as the bluebacks do. Next slide, thank you. Okay, so here are the numbers. This is what it looks like. These have been updated slightly since the last time I pulled this table together. You'll have to wait for the surprise from Sean's paper, but the trends don't change and the numbers don't change all that much either. Some folks may have not seen the updated Canadian numbers that'll be in that work. Big picture takeaways here is we're talking about minimally about 65% of alewife habitats that is affected by dams and fresh water in some capacity. That's in the Mid-Atlantic region. Again, the density of dams not quite as high there for them. As you move up the coast into southern New England, you're talking about 83% of their habitats above at least 1 dam. And then by the time you hit northern New England, about 90% of the habitats upstream of a dam. Those numbers actually jive pretty well with some analyses that Adrian Jordan and others were involved with a few years ago. And you know, one kind of important thing to recall from that paper and from our experience with this exercise is that a lot of those dams aren't mapped very effectively either for GIS softwares. Blueback herring, the table on the bottom is a little bit more of a mixed bag. Again, in the southern extent of their range, and they're not seeing as much habitat loss. They don't penetrate quite as far inland in those systems, or at least that's our thinking about their habitat right now. So, you know, 25–30% habitat loss down in the southern Atlantic and mid-Atlantic region. But although those percentages are smaller, the magnitude of loss is very high because there's a lot of habitat there. Again, those get a little bit higher as we include the Susquehanna and the Delaware River and those numbers as well, and the magnitudes of those, not the dams, because the Delaware doesn't have many of those. As you work up through southern to mid-coastal and northern New England, you see a very similar pattern that we saw with alewife, anywhere from 50% or more. And then by the time we get into northern New England, again, we're sitting at about 90% of the habitat above the first dam. Go ahead to the next slide, please. So this is a quick take on some coastwide abundances. This is really, you know, thrilling to be able to present figures that have potential of 400 million fish. And in fact, as we've included Canada and other habitats around the coast, this number is now pushing over 1 billion adult fish, which is just mind-blowing to think about the possibility. This is the thing that gets me so excited about these species, right? Unfortunately, we're not sitting probably very close to that 350 or so million fish right now. And so as we take our eye across the x-axis, you've got your no passage and no dam scenarios on either end. And much as was the case with American shad, we expect that the current levels of fish passage that we have around the coast are likely not moving the needle very far forward, should we say. Go ahead to the next slide. You can see similar trend there if you break that out regionally for alewife. Again, you know, those magnitudes are different based on the amount of habitat available. Really interesting to me is that despite the larger amount of habitat proportionally that's obstructed upstream of dams in southern New England and northern New England, you might not expect as much bang for your buck in a couple of those regions under the current passage scenario as you see under the no passage scenario, for example, for the mid-Atlantic region. All things considered, the trend very much similar to the coastwide trends. Next slide, please. For blueback herring. Blueback herring not quite as staggering in numbers of projected spawners around the coast. Again, these numbers have bumped up considerably since the add of the Susquehanna and the Delaware and Canada to these data sets. But again, pretty modest increase under what we're thinking of as a current scenario for fish passage compared to either the no passage scenario or the no dams scenario. Next slide, please. Likewise, broken out by region here, so you can sit down and look at your home region if you're interested in it. Again, most of the habitat and then correspondingly most of the abundance of these fish, most of the population size, and the southern Atlantic and mid-Atlantic regions, proportionally more dams in southern New England to Canada, really southern New England to northern New England, but lower magnitude overall of abundances therein. Next slide, please. Okay, so let's summarize this before we talk about where we're heading next. This is really exciting, and I really can't get over it. We've got functional coastwide models for these populations now. The tools, these R tools, are all driven by stock assessment data. They're peer reviewed in that regard. The shad work has been peer reviewed. The models themselves have now undergone peer review through Software Journal, and Sean's got the river herring models in review now. We've done some really fancy footwork in the last 6 months or so since the last time I've talked to most of you, and we've gone from the 51 Huck 10 units or Huck 12 units, whatever we were working with for the stock assessment. We've broken those all down now into coastal outlet systems so that somebody could sit down and play with their favorite river that's part of a larger stock and have a little mini model for those. All told, I think there's somewhere around 160 American shad population models, 220 or 230 blueback herring and alewife models each. And if that's not sufficient for the imagination, folks can now make their own rivers and their own habitat, so you can simulate an arbitrary population while using the regionally specific stock assessment data. So that one's just absolutely terribly exciting for me. The rest of this, you know, is maybe no brainer after seeing a few of those graphs, but we've got a large amount of spawning habitat and rearing habitat upstream of at least one dam for both of these species. This varies regionally, right, between the southern regions and the northern genetic reporting regions. But regardless of that, we are seeing that as anticipated, population productivity is following that proportionally. I mean, that's a matter of how the models work, right? But also that these current rates are likely ineffective for where we want to be heading for many of these populations. So where are we heading next? Next slide, please. We are now working on trying to make these tools more accessible. It was maybe like a month or two ago, and I told a couple people this might never happen, and then all of a sudden I got this amazing computer science intern, and he's now building for his internship a nice shiny app that folks can sit down and play with. This shiny app is more or less set to go, and it's as nice as any that I've made, but he's really good at this stuff, and he's looking to include include it. And every single one of those 200 some odd rivers for each of the species can be selected and population models run. This one's just showing the Abigadasset because it's both fun to say and alphabetically first for alewife. And there is Bryce there in the bottom right corner of his first alewife graph. So I'm really excited about this. He's going to be done with his internship this December, and we should have a preliminary product out to folks that they can actually sit down and play with without having to write R code, which I think is pretty powerful. So then finally, the last slide is just a big thank you to everyone who's been helping out, folks who've provided data, who've provided expertise. ASMSC's been incredible to us in letting us play along and build these tools alongside of the assessments. NOAA's been incredible to us through financial contributions to this work now for about a decade. And then folks like Maine DMR, DEC, close collaborators at those agencies who have been pushing us along the way. Of course, UMaine and the co-op units, Josie, and then a whole bunch of folks from Fish and Wildlife Service, as well as our own little SUNY Oneonta there in upstate New York. And with that, I'm all done.
Thanks very much, Dan. Are there any questions? Please start. Kevin and then Jonathan.
Hey, Dan. Awesome work as always here. Always love seeing your presentations here. I think we kind of talked about this before, but I'm not really sure where you've landed on it. You know, looking at, if we wanted to look at a model like this for, say, the Connecticut River, I'm trying to wrap my head around, you know, what we can do with a model, you know, where the Enfield Dam is now gone, so that was the first dam on the system. So we just have Holyoke, and our numbers have just continued to drop since the 80s. So we were up to like 630,000 fish in the mid 80s. Now we're down to a few hundred fish. So when you're at less than 1% of where you were pre, not restoration, because I wouldn't say we're fully restored there, but as we're doing this work to open up tributaries and fix fishways, remove dams, if we're dealing with populations that continue to go down, you know, kind of what as managers should we be doing where it's, you know, we put a lot of time and resources into looking at dams and the effect of dams, but you know, I think we've seen that figure once today that shows the historic landings of river herring coast-wide, and you can see those numbers were massive in the 50s and 60s. But there was a lot of dams. All these dams essentially were already built, and even Conowingo, I think, was like 1928. So at the time, with really high landings of river herring, all these dams were in place. So it's, you know, we're dealing with such small numbers of river herring now that it's awesome to see what could be produced if we could get them above all these systems. But where we are getting them above, we're struggling to get these fish back. So I'm just trying to think of what, you know, how as a manager to utilize these to tell me what's going to happen if we have other, like, is there a way to put other factors into these models? Because this stuff's obviously really helpful, especially if we're sitting down with an NGO that's getting grant money to remove dams. But it's really hard to then 10 years later have even less fish coming back and pass that straight face test when you've put like $15 million into removing dams. I'm just trying to think of how, you know, is there a way to pull in something like that, some other inputs into this model?
Sure is.
Perfect.
I mean, yes.
Yeah, at some point I'd love to do that with you.
You can, I mean, it's built in already. They're already, you know, functionally you could treat any one of those things as a fishery harvest rate, right? Dams included. Dams are separated out right now because that's where we pulled our funding from to be able to assess these things in the first place. But there are also a variety of fishing mortality rates. You could play with things like ALIA effects through the carrying capacity, the density independent or density dependent parameters. There are data out there now about climate change impacts on fish growth, et cetera, and so at the core are the stock assessment data, but I totally hear you. They're not going to fit every situation, right? And the Connecticut River and maybe more correctly some of the Connecticut tribs and the sort of southern New England area in general is a bit of a head scratcher for these kinds of reasons, right? You pull out a couple dams up in Maine and you're dealing with millions of river herring, and you do the same thing down in coastal southern New England, and you don't see the same result. And I'm not sure biologically kind of what the disconnect is there between that and what we would predict, right? And this is something you and I have spent some time talking about as well. But you absolutely can go into those models and tweak them and add those kinds of rates. Most of them are already options that users can modify in there. And the next big steps will be really kind of taking the hood latches off. We're trying to do it little by little. Right now, the R package is so data heavy that it can't even be submitted to CRAN. It's got like 15 megabyte habitat data sets. My hope is little by little to work that to a point where people could come in and punch in all the values that they want to use, and they could kind of come up with a custom scenario. So there's the kind of two questions is one, yes, you can absolutely chuck stuff at that. And try to hit those big numbers, even bigger numbers, right? That predated the Conowingo Dam and all those other dams, because those were big numbers, but they were still dropping the bucket to what was really out there before, so I don't want to also get sucked away thinking about, the 1950s as the heyday for river herring either, because I'm guessing they had it pretty bad by then. But you could go out and hit it with those kinds of scenarios. I've been talking with Lars Hammer up in Maine about some of this as well, and it seems like with the big connected river systems, we do a pretty good job of approximating restoration scenarios. So like the Penobscot restoration, some of the Kennebec Dam removals and the Sebasticook, we're hitting numbers that make a lot of sense for those. For the Connecticut, we're hitting numbers that make a lot of sense in the models, but you're not seeing that on the ground, right?
Yeah, exactly.
So there's also kind of the other piece, that's the biological conundrum of what the heck is going on there, right? And I think a lot of us are, especially academically, kind of scratching our heads similarly about the Hudson and Delaware, right? Where we don't know, one, in those systems just how many fish we have, but I think the general sort of suspicion is that maybe it's not as many as we should have, right? So it's kind of a long-winded way of saying, yeah, there are options in there to do it, and to I don't know what's causing all that funniness.
No, I think I where my mind is at with that. I just wanted to see if there was a way to put my thoughts into this. So thank you, I appreciate it. I'll definitely be reaching out to you at some point soon here. Thank you.
Hey, Dan, Jonathan here. Appreciate you coming back to present at the forum. My question is kind of along the same lines as Kevin's, but maybe a bit more specific. So I appreciate the scintillating preview of the R Shiny package. I started sort of beating my head against the R, writing code in R again, which I hadn't done in a while. And this is certainly going to make me slow that roll a little bit until I see what comes out in that Shiny package. But was curious, and I didn't get a good look at the screen when there was a slide sort of previewing it, but was curious if it was customizable. You anticipate the R package or the Shiny app being customizable to the extent that you could, say, go in and manipulate survival parameters of dams or passage parameters at particular dams, and look at scenarios under which, say, passage was improved, or and certainly both upstream and downstream, if it was going to be like to that level of detail, or if you're still going to have to write your own code essentially to describe potential outcomes from, say, things like passage improvement.
Yeah, those things are all going to be wrapped up into it. So there's two pieces to that. One, yes, the fishery harvest rates, the dam passage, sex ratios, carrying capacity parameters, those can all be tweaked by the user in the Shiny interface. The other piece of that question, the dam specific stuff, right now, as far as I recall, I could be wrong, but right now the only dam specific stuff that works 100% correctly is for systems without branches, right? Which is none of our rivers. It would be like the Connecticut without the Deerfield or West, or any of those, right? And that's somewhat useful. It's not as useful as it could be. And so the next piece that we're going to be working on in terms of the math and the actual computing is to implement homing and straying matrices into the under the hood part of the R package that would allow people to use dam specific survival passage and survival rates. But yes, you can go in and you can punch in your own. It's just that if the river is more than a straight line, then it's watershed scale dam passage. So, 30% upstream at all of the dams. That said, the last piece that Bryce is working on for the Shiny app is so that is a toggle so folks can actually enter their own river system. And we've been playing around with this already. So I think with Lars, I was messing around and we went and made like a Passadumkeag model for Lowell Tannery way up in the Penobscot system. And we did the same thing in the Hudson Valley. There's a grad student who is working on assessing productivity of some of the tributaries there. And we've split the tributaries out as separate models now. And so that's the last piece of functionality I'm hoping that Bryce can implement in the Shiny package before we kind of send that out to the world. It's a stopgap. It's not a perfect fix yet, but I need a little bit more time to work out the matrix math on the homing and straying in order to make it manageable for users as well.
Thanks. It's exciting. Appreciate your work on the development of that Shiny app.
Thanks again, Dan. If there are there any last questions or comments? Those have gone up right until, basically up until 4 o'clock. If there are no further questions, then I think we can wrap up. Oh, Wilson, go ahead.
Yeah, I've got one more that I thought of for Shannon, if she's still on here. But I did want to thank Dan for that great presentation. Not sure I understand it all, but I know plenty of people do, and it sounds like a really important piece of work. So thank you. So is Shannon still on?
I am.
Oh, okay. Well, here's the question. So going back to my earlier question, with a little bit of a tweak to it, given that you have a couple of decades of data there, did you, or would it be possible for you to look at to see whether there was any change in temperature through time in those three river systems, and whether or not your center of distribution for the may have shifted at all relative to temperature? And my thinking is that, okay, if we're seeing the average temperatures in these systems increase in conjunction with climate change, is there a possibility that the juvenile river herring are hanging around longer in the rivers just because they're staying warmer longer, and they might be increasing the growing season, or not? And I don't know whether that's something you could tell by looking at to see whether there's any change from what, 1996 when you started. Just a thought.
Yeah, no, that's a thing. Oh, go ahead. Oh, sorry, thought somebody else was jumping in there. Thank you. That's a great question. So we indirectly kind of approached that in a couple of different ways. Definitely, feel free to check out our paper because I'll try and go through this pretty quick, not to take up too much time. We did look at those mean centers of mass in warm years compared to cool years, and we also did that for wet years compared to dry years. And we saw, well, there was definitely mean centers of mass were further up estuary in wet years compared to dry years. Cool versus warm years, we didn't see a huge difference in those spatial mean centers. Yeah, that's kind of the long and short of it, quickly summarized.
Okay, thanks. Yeah, I'll read the paper. I'm glad you took a look at it. Thank you.
Yeah, thank you.
Jonathan, do you have anything else to add? Oh, got it. Thanks. Okay, well, if there are no more questions, I just want to give another big thank you to all of our presenters. Well, thank you to everybody who participated. Thank you for joining this afternoon. I will be sending out a follow-up email at some point soon with at least the links for the New England Mini-Land Council meetings coming up, as we talked about earlier, as well as the Manomet link and the link to the old presentation or one of the recent presentations that we've had at the forum on that as well. If we can get it done in time, because I know the public comment has a deadline, I would also include in that link or in that email the link to the YouTube recording of this if you want to share it to people who couldn't make it today, or if you can only make it for part of the day. If not, I'll send that out in a separate email. I'll make sure to follow up with that for sure. Other than that, Jonathan and I, I'm sure at some point, after the shutdown, we'll discuss the dates for 2026, and so look out for that email as well when we get the new GoToWebinar link set up for the dates next year. And with that, just a big thank you again to everybody, and I appreciate it, and have a good rest of your day. Thank you.